Come on, everybody. You are on the channel for English for Biology majors, which is brought to you by Tzu Chi University's Susan Ye. We hope after you learn this class, you will think biology is fun and to read biology book is piece of cake. Let's go, yay! Hello everyone. Today we are going to learn the third part of lesson 16, which is still frequently used prefix and suffixes. But first we need to review the second part. Remember, for fear, there's several prefixes and suffixes. Phobo, P-H-O-B-O, and phobia, P-H-O-B-I-A, this is a suffix, and also the suffix phobic, P-H-O-B-I-C. To describe things that are the same, we have several prefixes. Homo, H-O-M-O, and ipsi, I-P-S-I, and iso, I-S-O, and tauto, T-A-U-T-O. To describe things are different. We have hetero, H-E-T-E-R-O. Now let's move to the third part of lesson 16. First, what we are going to learn is to describe things are good or well or true. We have this prefix, EU, U. Our example for EU is eukaryote. Eukaryote is an organism whose cells have a true nucleus, which means the nucleus is bounded by a nuclear membrane within which lies the chromosome and the associated proteins, and which exhibits mitosis. And also it contains membrane-bound compartment, which is organelle, in which the cellular functions are performed, and have a cytoskeleton that provides mechanical support for the cell. Members of eukaryote family includes plants, animals, fungi, and the protists. They are all eukaryote. When we want to describe things that are false, we have this prefix, pseudo, P-S-E-U-D-O, or pseudo. Our example is pseudoscience. Pseudoscience is a system of theories, assumptions, and methods erroneously regarded as scientific. It is a claim, a belief, or a practice which is presented as scientific, but actually it does not adhere to a valid scientific method. Pseudoscience lacks supporting evidence or plausibility, and also it cannot be reliably tested or otherwise lacks scientific status. Like there's a saying that cut onions can prevent influenza, which is pseudoscience. And also, several years ago, there is a paper talking about that goodwill will change the structure of water crystals, but actually later nobody can retest that theory, and the so-called research team later admit that they falsified the evidence, all of which are regarded as pseudoscience. When we want to talk about eat, we have several prefix and suffix, including phago, P-H-A-G-O, this is a prefix, and then three suffixes, phagia, P-H-A-G-I-A, phagy, P-H-A-G-Y, and the phage, phage. Phage is usually being used to describe an organism, or a cell. And then phagy is used to describe a process, a phenomenon. And phagia also is used to describe a phenomenon, but more like an abnormal type of phenomenon. Let's see our example. For phago, our example is phagocytosis. Phagocytosis is endocytosis of particular material, such as microorganisms. Like in this picture, the macrophage is engulfing, is try to eat the bacillus, and because bacillus is a particulate material, so this process is phagocytosis. Not only for microorganisms, also like fragment, things that have shape, that are solid, then we use the term phagocytosis. And after it's swallowed, the material is taken into the cell in membrane-bound vesicles, which called phagosomes or phagosomes, that originate as pinch-off invaginations of the plasma membrane. Phagosomes fuse with lysosomes, forming phagolysosomes, in which the engulfed material is killed and digested. That's phagocytosis. And our example for phagia, the suffix phagia is dysphagia. The prefix dys means pain, not being able to do it. So dysphagia is difficulty in swallowing. Like the bird in this picture, it's trying to swallow something, but if it has dysphagia, then it will be really, really hard for it to do it. For suffix phagy, phagy, our example is geophagy. Geophagy. What is geophagy? It is also called geophagia. The prefix geo means soil. Geophagy is the habit of eating clay or eating earth, like parrots. Some parrots will eat clay or soil, so we can say that it has geophagy. In the world, in some areas, people will eat mud cake, and that is also one form of geophagy. And you can see this picture from Haiti. People even made mud cake and sell it. They only made mud cake from some certain areas in the river. They said the mud there is having some nutrition that most people lack. So people will buy it and bring it home to use it as a nutrient supplement. This is geophagy. For phage, phage, our example is bacteriophage. Bacteriophage is a virus, like in this picture, that likes bacteria. It injects its DNA into the bacteria. When its replication is completed, it will lyse the bacteria, release hundreds of bacteriophages. For phage, we have one more example, macrophage. Macrophage, like we show in this picture, and also in the previous example, when we are talking about phagocytosis, we also see a macrophage. Macrophage are any of the many forms of mononuclear phagocytes found in tissues. They arise from hematopoietic stem cells in the bone marrow, which develop according to the stages of the monocytic series until they are monocytes. And then the monocyte enter the blood, circulate, for about 40 hours and subsequently enter tissues where they increase in size and the phagocytic activity and also the lysosomal enzyme content. They officially become macrophages. Now let's review what we learned. For eat, we have one prefix, phago, P-H-A-G-O, and then three suffixes, phagia, P-H-A-G-I-A, and phage, P-H-A-G-I, and phage, P-H-A-G-E. When we want to describe the constituent substance of the cell, there's two prefix and suffix to use. For suffix is plasm, P-L-A-S-M, and for prefix is plasmo, P-L-A-S-M-O. For the suffix plasm, P-L-A-S-M, our example is cytoplasm. Cytoplasm is the protoplasm of a cell exclusive of that of nucleus. It does not include nucleus. Cytoplasm consists of a continuous aqueous solution. Now, the 11 in this picture, the part 11 in this picture, this is called cytosol. And then it also includes the organelles and inclusion suspended in it. All the organelles like mitochondria, centrioles, vesicles, and ER, Golgi. These together with the cytosol is called cytoplasm. For plasmo, our example is Plasmodium. Plasmodium is a protozoan of the genus Plasmodium. Plasmodium, actually, they are the malarial parasites. Now, as shown in this picture, this is one of them. This is another one. They belong to a genus of coccidian protozoa of the suborder Hemosporina. They cause malaria if we are infected. Let's review what we learned. For constituent substance of the cell, there's one suffix, plasm, P-L-A-S-M, and one prefix, plasmo, P-L-A-S-M-O. When we want to talk about enzyme or fermentation, now fermentation is also an enzymatic process. We have one prefix and two suffix to use. The prefix is zymo, Z-Y-M-O, and the suffix is Z-Y-M-E, zyme, and the A-S-E, ase. Our example for zymo is zymosan. This is a picture of the structure of zymosan. Zymosan is a mixture of polysaccharides, proteins, and ash of variable concentration derived from cell walls of the entire cell of yeast. And it is being used to absorb the C3 component of the complement. Therefore, it is used for assaying propyridine. Propyridine is a non-immunoglobulin gamma globulin component of the alternative pathway of complement activation, which you will learn in the course of immunology. And zymosan is made from the cell wall of yeast, and it's used to absorb part of the complement system. And for the suffix zyme, Z-Y-M-E, our example is ribozyme. Ribozyme, as shown in this picture, is a hammerhead ribozyme, which is an RNA molecule with catalytic activity. There are several classes of different ribozymes exist, and the reaction they catalyze may be intramolecular, like self-splicing. They intron self-splicing involves some ribozyme activity, or it may be intermolecular, like maturation of tRNA molecule. It requires enzyme to do it, and that enzyme is a ribozyme. For ribozyme, the secondary and the tertiary structure is very important for its activity. And then for ASE, the suffix ASE, we need to talk about the general rule of enzyme nomenclature, how to name your enzyme. First, all of the enzyme must come with the suffix ASE, denoting it is an enzyme. And then the first part of the name include whether it's substrate or its product. And also, it includes the reaction type. Let's use an example to explain it. The example is DNA polymerase. DNA polymerase, as shown in this picture, these two guys, DNA polymerase alpha, which is in charge of replicating DNA. The name of DNA polymerase, we can actually separate them to three parts. The first part is the ASE suffix, meaning it's an enzyme. And then the second part, DNA, which is the substrate of this enzyme and also the product. Now, the substrate is a shorter DNA and the product is a longer DNA, but they are both deoxyribonucleic acid. And then the third part, polymer. Polymer is denoting the type of reaction, which is polymerization. So the name DNA polymerase, actually it means an enzyme that is in charge of polymerizing DNA. Okay, now understand how to name your enzyme. Now we finish the third part of lesson 16. Let's review what we've learned this time. For good and well, we have EU. And for false, we have pseudo or pseudo, P-S-E-U-D-O. For eat, we have phago, P-H-A-G-O, this is a prefix, and phagia, P-H-A-G-I-A, phagy, P-H-A-G-Y, and phage, P-H-A-G-E. For constituent substance of cell, we have plasm, P-L-A-S-M, which is the suffix, and plasmo, P-L-A-S-M-O. For enzyme or fermentation, we have zymo, Z-Y-M-O, this is a prefix, and also two suffix, zyme, Z-Y-M-E, and ase, A-S-E. Now we've finished what we need to learn this time. Next time when we come back, we'll move to the fourth part of lesson 16. See ya! English for Biology Major is brought to you by TCU's English for Biology Major course team. If you have any questions, don't forget to visit our FB fans page. The website is here, and you can also scan the QR code to reach to our FB fans page. This course is sponsored by Centers for Faculty Development and Instructional Resources of TCU.